Okay, welcome everybody. I'm the chair of Mahafuza's Defense Committee, so I will just very briefly say welcome. And Mahafuza will give her defense, and at the end of that, everyone except for the members of the committee are welcome to ask questions. And after that, we will go into a closed door session.
Recording in progress.
And after that, we'll go into a closed door session with the committee, where we will further grow Mahafuza on your thesis. So, welcome to your defense, Mahafuza, and now I'll turn it over to John. Okay. Professor.
All right, well, thanks all. I'm really happy that you're here. I'm going to stop letting students graduate because my pulse always quivers the week beforehand somehow. But I'm really excited to be here to introduce Mahafuza to you. Mahafuza has been in our group for a while now and has done some really amazing things and a lot more things than she's going to talk about today, which I also very appreciate. So I want to tell you a little bit about who Mahafuza is as a person. Now, she started as a clinician, which I think is pretty amazing. So if something happens today, like we have a clinician in the room. So Mahafuza was a clinician in Bangladesh before coming to the States and deciding to make that transition to doing science, which of course we're all extremely happy that she's done. But I can imagine that is a difficult transition. I think Mahafuza talks about that transition in an interview that's on YouTube, but I didn't understand any of it, and ChatGPT failed the translation. But anyway, I think it's been amazing. Mahafuza came first to the U.S. and kind of much like my former advisor, Tig Rainey, when he decided to come to the U.S., he's like, if I'm going to go to America, I'm going to go to Texas. And so Mahafuza started her scientific career at Texas, where she worked with Jenny and did some really cool work looking at neurogenesis and an epilepsy model, and actually has several authorships from that time, including in Nature Communication manuscript. So, like I've done several of these introductions, and I kind of wanted to model it to the person. I really thought Mahafuza deserves like a Margaret Vizard introduction. So I started thinking about like the characteristics that I really appreciate about Mahafuza in the lab. And I think the first word that came to mind was her dedication to pretty much everything. Like I was first thinking about Mahafuza is so dedicated to the lab, and she's been such an integral part of the lab, and willing to do most anything that we talk about scientifically in the lab, but also dedicated to her science. I mean, as you'll see, she really was critical in turning our rat lab into now, certainly for a while primarily a mouse lab. Now we're kind of doing both, but I think introducing that model and working with some of these new mouse models that we have, she's been integral in introducing that to the lab, which is really powerful and really pretty amazing. So Mahafuza has been dedicated both to us as a group and also to the science that she's going to be talking about today. I don't think most people get to this point without having resilience as a key trait to doing science, to getting to a PhD. Mahafuza certainly, I think, characterizes that resilience in a way that not all of us, have to do in terms of not just navigating the science. And I know Mahafuza has had some tough challenges in terms of the science, because I get a lot of kooky ideas that make me know that it doesn't work. And certainly Mahafuza's never said no. Like, we've always had the conversation. She's always been energetic and going in and jumping in to these experiences also. But in terms of also navigating how to do science in a completely different culture, in a new country, perhaps with a language that isn't necessarily your primary language. I have to say, in terms of following Mahafuza's career, I am the son of someone who did that, and I've always been really appreciative of that, of my own mother. So seeing that in Mahafuza has been really endearing in terms of her ability to overcome a lot of things to get to this point. And you have to be resilient to work with such a motley crew as we have on the fourth floor of Dewey. You also have to be patient. I'm not sure that this picture represents patience or just cold, but it looks like patience to me. Mahafuza has certainly been extraordinarily patient, both with me as a mentor, with the science as it works and sometimes doesn't work with everything. So I'm thinking about all of these characteristics. These are just amazing characteristics, but they really are characteristics of someone who has another job, another very important job. And I don't know Mahafuza as a mother, but I can't imagine someone who might be a better one in terms of the characteristics that Mahafuza has. So I really do appreciate that. There is another characteristic that could be mom-like, but is not specifically mom-like, that I think Mahafuza characterizes, which I think will take you far in your scientific career, and that is curiosity. Mahafuza has always been willing to ask questions about everything, including, of course, the science, but also including the culture and the scientific culture and navigating the science. And I've always really appreciated those conversations and the honesty of which you ask questions, and are always looking for always looking ahead and always looking for the challenges and to meet them. So Mahafuza has done a lot here, as I've said. Won awards, given a lot of presentations that you've seen, and it has been really grown a lot since I've seen it. Since her initial time in the lab, it's been pretty amazing. So I was thinking about, like, okay, well, you know, we do this thing in the lab. We're like, oh, what fictional character does each person represent? And I didn't want to leave you without that. And so when I was thinking, like, I'm an older person now, so I was thinking about, like, okay, well, I need someone who is just really strong, resilient, perhaps survives when no one else, when all the bumbling men don't survive, but also has parental qualities. And so I was thinking, oh, well, the first person that came to mind was Helen Ripley from Aliens. So I'm going to hold on to that, even though I have no idea who this is. So anyway, I'm so happy to introduce you to Mahafuza. I think this is going to be great, and look forward to a couple of minutes. Great.
Thank you so much, John, for an amazing introduction. I'd like to thank you everyone for coming to my television defense presentation. Today, I'm going to talk about the role of backbone receptors in the benefits of terminalis and anxiety. So we all know that stress response is important for survival, adaptation, and maintaining homeostasis. But chronic exposure to chronic stress is an important risk factor for anxiety disorders. Anxiety disorders is one of the most common psychological disorders affecting more than 40 million American adults, where 1 in 3 people are affected. Specifically during the pandemic, we saw a rise of anxiety disorders. According to the World Health Organization, there was a 25% increase in prevalence of anxiety and depression worldwide. Post-traumatic stress disorder, or PTSD, is also a psychological disorder developed by exposure to traumatic event or events and producing symptoms like nightmare, flashback, and severe anxiety. In 2013, American Psychiatric Association revised the diagnostic criteria of PTSD in their 5th edition of Diagnostic and Statistical Manual of Mental Disorders. And in the new category, PTSD is part of trauma and stress-related disorders. Previously, it was a part of anxiety disorders. Our lab, along with Russell lab, has shown that the blood level of pituitary adenine cyclase activating polypeptide, or PACAP, and its single nucleotide polymorphism in BAC1 receptor gene is associated with PTSD in women. They have also shown that this traumatized woman has increased dark enhanced startle. So these data, along with other data, suggested that PACAP is important for chronic stress and may play a role in anxiety-related behavior. So what is PACAP? PACAP is a pleiotropic peptide. Pleiotropy defined as one single gene that can produce more than two characteristics of phenotype. As PACAP is a pleiotropic peptide, it has several different functions. Importantly, neurodevelopment, neurotransmission, homeostasis, trophic support, self-proliferation, self-protection, so on. PACAP has been a really conserved neuropeptide. When it first discovered, it was discovered as a hypophyseal factor that can stimulate pituitary cells to secrete cyclic AMP. It is processed into PACAP27 and PACAP38 post-translationally. It has been conserved for 700 million years, which implies that PACAP is super important for biological function. It belongs to VIP secretin and glucagon peptide family. In central nervous system, PACAP38 is more common than PACAP27. So from these slides, I just wanted to give an important point, that is PACAP is very important for several different biological functions. The highest concentration of PACAP has been observed in hypothalamic region. Among hypothalamic region, most intense PACAP staining has been observed in medial hypothalamus compared to the lateral hypothalamus. Apart from hypothalamus, PACAP has been observed, PACAP cells and fibers have been observed in various extrahypothalamic region, such as cerebral cortex, hippocampus, where nucleus of start terminalis, amygdala, and parabrachial nucleus. PACAP specifically can bind to PAC1 receptor, but it can also bind to VPAC1 and VPAC2 receptors. These receptors are G protein coupled receptors. They belong to class B family of G protein coupled receptors. Those receptors have 7 transmembrane helical with long N-terminal domain. PAC1 receptors has various isoforms depending on 84 base pair amino acid of HIP and HOP acid. It is important because depending on the isoform, PAC1 can differentially activate GS or GQ and activate several signaling cascades. Importantly, one of the signaling cascade is through beta-arrestin mediated endocytosis of PAC1 receptor, which can produce prolonged signaling. PACAP is a key regulator of stress circuit. Stress circuit can be mediated by behavioral stress response, endocrine stress response, or autonomic stress response. And PACAP can play a role in all the stress response level and stress response level. PACAP can activate the paraventricular nucleus of the hypothalamus to secrete corticotropin releasing factor or CRF. CRF can stimulate pituitary cells to secrete adrenocorticotropin releasing factor or ACTH. ACTH can activate the adrenal cortex to release glucocorticoids. PACAP can also stimulate adrenal medulla to secrete catecholamine. These are important hormones for stress response. Lee Iden and his group has been suggested that there is 2 distinct PACAP circuits in the central nervous system for stress responding. One is via medial prefrontal cortex to PVN to to produce stress, endocrine stress response, and one is via parabrachyal nucleus to PNST and central nucleus of amygdala, which produce behavioral response of stress. So the important point here is PACAP is very important for stress responding, and it can modulate different levels of stress response. So our lab has previously shown that after repeated stress exposure, the PACAP transcript in the PNST can increase significantly. In this experiment, we use chronic stress paradigm, which I will discuss it later. After exposure to chronic stress for 7 days, male rats, the brain of the male rats are dissected and then look for the PACAP transcript using PCR, or polymerase chain reaction. And if we look at the graph A, we can see that the stress group in dorsal PNST showed significantly high PACAP transcript compared to the control group and other parts of the brain. C and D are the representative image of PACAP cells using in situ hybridization. If you look at E, we can see the number of PACAP cells in the PNST is significantly higher than the control group. So this data is suggesting that PACAP is important in the PNST following chronic stress. So now I'm going to talk about PNST. What is PNST? PNST is a complex heterogeneous limbic structure which is composed of collection of subnuclei. Most of the PNST cells are GABAergic, some with some glutamatergic projections. These PNST cells co-express different neuropeptides, importantly PACAP and CRF. PNST cells can respond to long duration aversive stimuli, which are temporally distant or unpredictable. Our lab also shown that by PACAP infusion into the PNST, we can produce anxiety-like behavior. In this experiment, male rats are cannulized bilaterally in the PNST and infused with different dose of PACAP and then measure the startle amplitude. If we look at the graph, you can see that PACAP 38 dose-dependently increased startle response in PNST, startle response in PNST. So, which suggested that PACAP in the PNST can produce anxiety-like behavior. So here I'm like, I want to recap. What I have shown you is that PACAP is important for stress responding, and PACAP in the PNST can produce anxiety-like behavior. So this angiogenic behavior in PNST is mediated by PAC1 receptor. Previous graduate student in our lab, Caroline Roman, has shown that angiogenic PNST PACAP signaling is mediated by PAC1 receptor. In this experiment, we have 3 groups of rats who are cannulized bilaterally in the PNST. The first group was infused with vehicle. The second group was infused with maxilillin, and the third group was infused with VIP. Maxilillin is a PAC1 selective agonist, and VIP is VPAC1 ligand. And she used a behavioral test called open field test, where when we put the rat into the box, if the rat spends more time in the periphery compared to the center, that produces anxiety-like behavior. So if we look at the graph A, we can see that the maxilillin group spends significantly less time in the center compared to the vehicle and the VIP group. And if we look at the graph B, we can see that the maxillary group spend significantly more time in the periphery compared to the vehicle and PID group, which suggested that BNST PACAP signaling is mediated by PAC1 receptor. She also showed that by blocking PACAP receptor, we can block anxiety-like behavior. In this experiment, there was two groups. One group was exposed to chronic parent stress paradigm, and one group was handled and stayed in the home care. Both groups got cannulated and to get chronic infusion of vehicle or PACAP 638, which is the PACAP receptor blocker, via osmotic minicum. She used elevated class maze to evaluate the anxiety-like behavior. So if we look at the repeated stressor group, the vehicle group spends significantly less time in the open arm compared to the control groups. And this behavior was blocked by chronic infusion of PACAP 638. If we look at the graph B, we can see that the stress groups lost significantly more weight compared to the other groups, and which was blocked by infusion of PACAP 638. So these data is suggesting that by blocking PACAP receptor, we can block anxiety-like behavior. PACAP 638 not only blocks PAC1 receptor, but it also blocks BPAC2 receptors. We wanted to target just PAC1 expressing neurons, so we decided to use PAC1 transgenic mice. In PAC1 transgenic mice, there is a Cre-recombinance enzyme which situates downstream to PAC1 promoter. This Cre-recombinance enzyme can interact with specific DNA sequence called LOXP site, and by interacting with LOXP site, we can modulate or express different fluorescent protein in PAC1 expressing neurons. I just wanted to highlight one thing, that we are targeting the cells that express PAC1 receptor. We are not modulating the PACAP receptor directly. We also used PACAP mice to trace the PACAP expressing neurons in the PNST. In this, these mice also have the Cre-recombinance enzyme downstream to PACAP, and by using LOXP site containing viral vector, we can express enteric fluorescent protein in just PACAP expressing neurons. So my specific aims are to determine the behavioral consequence of modulating endogenous BNST PAC1 expressing neurons. My second aim is to determine the projection pattern of BNST PACAP and PAC1 expressing neurons. And my third aim is to determine whether chronic stress altered the expression of BNST PAC1 expressing neurons. So my first aim has two sub-aims. First sub-aim is to determine the behavioral consequence of endogenously inactivating the PNST PAC1 expressing neurons, and my second sub-aim is to determine the behavioral consequence of endogenously activating PNST PAC1 expressing neurons. For this group of experiment, we use designer receptor exclusively activated by designer drug technique or drug technique, where we artificially insert G protein coupled receptor which can activate or inactivate by designer drug called clonazepine and oxide. For these, we use inhibitory dread or GI dread, excitatory dread or GQ dread, and control virus. we bilaterally inject the virus into the PNST and wait for 10 days for viral expression and surgical recovery. After that, we inject the mice with CNO parenterally and then wait for 30 minutes for its activation, and then place the mouse in the center of the plasmase. So plasmase is a behavioral test where we evaluate the anxiety-like behavior. So there's two closed arm and two open arm. If the mouse spend more time in the closed arm compared to open arm, that indicates anxiety-like behavior. So first, we use 3 milligram per kg CNO, and when we use that dose of CNO, we didn't see any difference in open arm time compared between the control group and the GI group. We also didn't see any difference in open arm entries compared to the control group and GI group. That's why we decided to increase the dose to 10 milligram. So when we increased the dose of CNO to 10 milligram, we saw that there's a significant difference of GI group to control group. So GI group spend significantly more time in the open arm compared to the control group, and also GI group made significantly more open arm entries compared to the control group, which suggested that by inhibiting PAC1 expressing neurons, we can reduce the anxiety-like behavior. We also wanted to look at the total locomotor activities and make sure there is no locomotor effect. For that, we measured the total crossing. When a mouse moves from one arm to another arm, that's one crossing. And when we look at the total crossing, we didn't see any difference between the control group and GI group, which suggested that there is no locomotor effect. This is a representative image of red GI viral expression in the PNST. So after that, we wanted to stimulate the PAC1 expressing neurons started with 3 milligram per kg CNO. When we did that, we didn't see any difference between the control group and GQ group in terms of open arm time. But we saw a significant difference where GQ group made significantly less open arm entries compared to the control group. But we wanted to increase the dose and see what happens. And when we increased the dose, we saw that the GQ group may spend significantly less time in the open arm compared to the control group, and the open arm entries are also significantly less in GQ group compared to the control group, which is suggesting that by stimulating PAC1 expressing neurons, we can produce anxiety-like behavior. So we also looked at the total crossing, and there was no locomotor effect that we observed between control group and GQ group. This is a viral expression, GQ viral expression in the BNST. So to reiterate, when we inhibit the PAC1 expressing neuron, we can reduce anxiety-like behavior. When we stimulate the PAC1 expressing neurons, we can produce anxiety-like behavior. So we wanted to make sure there is no off-target effect using 10 milligram per kg CNO, which is a little bit higher than normal dose. So for this experiment, we used wild-type mice. One group was injected with vehicle, and one group was injected with 10 milligram per kg CNO. And we didn't see any difference in in terms of open arm time, and we didn't see any difference in open arm entries, which suggested that 10 milligram per kg CNO doesn't have any after effect. For my second specific aim is to determine the projection pattern of BNST backup and back-on expressing neurons. For this, we used both back-1 and backup mice. We injected the backup mice in bilaterally in the BNST with AAV MGRI virus, and then with using back-1 mice, we injected AAV MGRI, also retrograde GFP virus. After injection, we wait for 10 days for viral expression and surgical recovery, and then we transcardially perfuse the animal and prepare the tissue for analysis. When we look at the backup cream mice and the BNST, we saw that backup expressing neurons both in dorsal and ventral of anterior commissure in lateral BNST, as well as in medial preoptic area. It is important because in the literature, researchers have shown that there is backup fibers in the BNST, backup projection fibers, but there was a controversy about the local source of BNST, and we have shown that there is local backup neurons in BNST. There are different morphological types of neuron in the BNST. You can see that there's a F indicates fusiform projection neurons, B indicates heterodendritic bicone neurons, and S indicates superficial spiny interneurons. In the anteriorly backup, BNST neurons project to nucleus accumbens, both in the shell and the core region of nucleus accumbens. Nucleus accumbens is important for aware and addiction-related behavior. In posterior BNST, we see that there is two distinct neuronal fiber going from stria terminalis and stria medullaris, and the projection from stria terminalis projects to amygdala, and from stria medullaris it projects to medial habenula. Amygdala is important for defense and behavior, defense and fear-related behavior, and medial habenula is important for depression-related behavior. This is a schematic showing where BNST backup neurons are projecting to from the projecting to. So we are also curious about the projection pattern of BNST back-on expressing neurons. And we see the first picture is showing the back-on expressing neurons in the BNST. We see that these neurons project to lateral habenula, also in nucleus accumbens. Lateral habenula is important for depression-related behavior and nicotine withdrawal. Nucleus accumbens is important for addiction and reward-related behavior, as I mentioned before. These back-1 expressing neurons also project to amygdala, specifically in central nucleus of amygdala. And amygdala is important for defensive-related behavior, fear-related behavior, also emotional component of pain-related behavior. Lastly, back-1 expressing neurons projects to lateral hypothalamus, substantia nigra, microcellular tegmental area. So lateral hypothalamus is known for metabolism and feeding related behavior. Substantia nigra is important for motor planning, reward-related behavior, and addiction-related behavior. Microcellular tegmental nucleus is known for sleep, wake, and motor function. This is a schematic showing you the areas that PAC1 BNST cells are projecting. So next, we decided to do a retrograde tracing of PAC1 expressing neurons. There are a few reasons we did that. One is because we have the opportunity and we are curious to know where these fibers are coming from. Also, our lab has shown that before that backup begets backup, and so we went to see if that is true for PAC1 expressing neurons. So the first image we can see this, there are fibers coming into VNST, PAC1 expressing neuronal fiber. The cell bodies we saw that is coming from lateral habenula as well as thalamus nucleus. As I mentioned before, lateral habenula is known for depression and nicotine withdrawal related behavior, and thalamus is a relay center for processing sensory input. We also observed that the cells are coming from cingulate cortex and nucleus accumbens. Cingulate cortex is important for reward and punishment related behavior, and nucleus accumbens is important for addiction and reward related behavior. We have observed this. There's a lot of PAC1 cells are projecting from basolateral amygdala and hypothalamic nucleus. Basolateral amygdala is a part of amygdala which is important for fear-related behavior, and hypothalamus is important for metabolism and feeding-related behavior. Lastly, we have seen the PAC1 cells are projecting from striatum and substantia nigra. Striatum is important for decision making, and substantia nigra is important for motor planning and addiction related behavior. This is the schematic showing the areas that PAC1 neurons are projecting from VNST. Now, my third aim is to determine whether chronic stress alters the expression of VNST PAC1 expressing neurons. For this, we used red GQ virus and control virus. We injected the virus bilaterally into the VNST and wait for 10 days. After 10 days, one group stays in home cage and get handled every day, and the other group are exposed to chronic pharynx test paradigm for 14 days. After 14 days, the other mice are injected with CNO 30 minutes before the plasmase experiment, and after that we do the elevated plasmase experiment. As I mentioned before, it is a behavioral test to evaluate anxiety-like behavior. If the mouse spend more time in the enclosed arm compared to the open arm, that suggests that anxiety-like behavior. So, chronic stress paradigm, we use 5 different mild stressors. First one is oscillation. We put the mouse in a moving platform for 30 minutes. Then we use swim as a stressor. We put the mouse in a bucket for 5 minutes. And then we use foot shock. Yes, the mouse gets 2 foot shock with 5 minutes interval. And then restrain, 1 hour of restrain and a restrain 2. And that is the we put put the mouse in a high platform for 30 minutes. These stressors are different from each other to prevent habituation, and we do this for 14 days for this experiment. When we looked at the weight change, we saw that the stress group lose significant weight compared to the control group, which suggested that the stress has an effect on the weight gain. And we looked at the anxiety-like behavior. We didn't see, what we saw is interesting and unexpected. If you look at the time spent in the open arm, the first graph, the stress group spent more time in the open arm than the non-stress group. And also the stress group made more open arm entries compared to the non-stress group. We have few thoughts about it. One is this is maybe a strain difference. I have used previously back-gap mice and saw stress response and anxiety-like behavior. So though we backcross the back-1 pre-mice with C57 mice, there may be a genetic difference that is maybe causing the stress, the anxiety-like behavior that we are seeing is more hyperactivity than less open arm entries or spending time in the open arms. We also looked at the locomotor effect, and we didn't see any locomotor effect between non-stress and stress group. So we decided to look at other stress or anxiety related behavior. So we looked at rearing, grooming, and freezing behavior. So when we looked at the rearing behavior, there was no main effect of stress, and we didn't see any main effect of dread GQ, and no interaction. In freezing behavior, we also didn't see any main effect of stress or main effect of dread GQ or any interaction. So lastly, we looked at grooming behavior, and we didn't see any difference in main effect of stress or main effect of dread GQ or interaction. So there are a couple of reasons that I mentioned before. Maybe there is a cohort effect, there is a strain effect, or the oval nucleus that we always thought is the main center for anxiety-like behavior is not as we thought in this strain of mice. But we can discuss it later. So as conclusion, we can say that backbone expressing neurons in the PNST play a role in anxiety and stress responding, which is consistent with our prior work, implicating PNST back-gap release and signaling stress responding. PNST back-gap and back-1 neuronal fibers have distance projection to other limbic areas of the brain. We do see a chronic stress effect on weight change in back-1 pre-mice. PNST back-gap and back-on projections may coordinate the behavioral response to chronic stress, including anxiety-related behavior, pain, migraine, impaired learning, memory, and relapse from ambition. For future direction, we wanted to do the same experiment using female back-1 pre-mice. We want to look at the behavioral consequence of modulating endogenous back-1 expressing neurons. We also were curious about the neuropeptide and neurotransmitter releasing from back-1 expressing neurons. We wanted to look at if we modulate the projection from PNST to nucleus accumbens of BAC1 expressing neurons, what are the behavioral consequences. We also wanted to look at the projection from BNSG to lateral half annula and modulate this projection and see what are the behavioral consequences of this. So, thank you so much. I'd like to thank my advisor, John Hammock. Without you, I would not be here today. And I was thinking about when I started my PhD, I thought, oh, I can do it. It'll be easy because I have life experience. I have done real life jobs and it'll be fine. And then I started my PhD. There are days, weeks that are like, what am I doing? I don't know. Nothing makes sense. And then I talk to John and I feel better. It's like you can make negative experience, flip it, make it positive. I'm like, oh yeah, it makes sense. Yes, nothing like my experiment failed for the last 6 months, and it doesn't matter. I learned something from it. So anytime I feel stressed or depressed, I talk to John and I instantly feel better. I also wanted to mention that because I'm an international student, I didn't have family around. When my first time my kid was very sick and I had to take her to the emergency, I feel very helpless because I don't have anyone. Then I texted John, I was like, I feel instantly, he just replied to me and I feel like, yeah, I have someone that I can rely on and I feel better. And so, thank you, John, and I'm super proud that I'm your student and you're my mentor. And I'd like to thank my committee members. Thank you. Donna, you have been my inspiration. Thank you, Travis. Thank you for helping me to navigate my job situation. Thank you, Tony, for being there for me from the first day. I'd like to thank my lab members, Nate, Sam, Emma, Jake. And I like to thank all the undergrad that helped me for my experiments. And I like to thank my family, my friends, I lost my dad in 2011, but I think he will be really happy if he was here. My siblings, I like to thank my friend Tasmin, Faria and Nusrat for being my support system. And I'd like to thank Chris and my daughters, Jane and Rosa. Thank you. Any questions?
Any questions?
You talked about the chronic variant stress system, and that didn't show effects in kind of psychokinetic differences might be the cause. What about acute? Have you tried anything with an acute stressor that maybe also parallels with human experiences? PTSD can be caused by a singular incident or a chronic incident.
That's a very good suggestion. I have not tried acute stress.
Great, nice story. It's so nice to see you, wrapping things up here. I was curious about the nucleus accumbens. You mentioned that could be a future target, and just, curious if you were going to stay here another year or 2 or 3 and going to pursue that. What would that look like? Like, what kind of behaviors would you target with those PAC1 projecting neurons? I think that's what you said.
Yeah. So there are some literature that back up in nucleus accumbens as related to like reward related behavior, that they have shown that back backup. Injection in the nucleus accumbens can reduce palatable food consumption. And I can do the same experiment with using PAC1 premise and see if that's what's happening with the palatable food, because it's like a proxy for reward related behavior. So that's one of my ideas.
Any other questions?
Is that okay?
Yeah,
Can I
of
ask a
course,
question?
yes.
I know you mentioned that you had some frustrations and you had to keep going. What would you want Jane and Rosa to take away from your experience?
I want them to know that there is always, it's like how you look at things. You can look at positively, negatively. It's okay to have your feelings and have your emotions and feel that, and then you get up and do your thing.
Thank you.
On that very wise note.
Yeah.
Thank you very much.